dedicated lecture is to cover the remaining bacterial groups and type species for the gram negatives. So let's now start with the spirochetes. The spirochetes, morphologically at the cell level, are corkscrew-like in appearance, and they have a very unique way of moving. They spin, and um, in terms of their motile or motility structure, they have flagella. They can have 6 or more endoflagella or a periplasmic flagellum and there could be axial filaments or axial fibrils, allowing it to carry out a spinning type of movement. These organisms are very fragile. If you remember from our discussion on laboratory diagnosis, so there, as an example, we talked about how important it is to keep their conditions or the collection conditions close to the ambient that supports the growth or that supports the viability of these microbial prokaryotes. They are microaerophilic, meaning they require a specific concentration of oxygen, about 2 to 10%. Um, they cannot survive in atmospheric oxygen concentration. So some examples of spirochete bacteria include Treponema pallidum, causative agent of syphilis, Borrelia burgdorferi, and Borrelia recurrentis, and Leptospira interrogans, which causes leptospirosis. So let's cover these type species. First is um, Treponema pallidum. Okay, so it's the etiologic agent of an STD, syphilis. It penetrates intact mucous membranes as it has the capacity to secrete hyaluronidase. It can degrade connective tissue material. They can also invade through epithelial abrasions, so any cut through the epithelial layer, um, they may be able to benefit or gain entry to the animal body, to the human body, via these abrasions. In the context of a mother and a child, neonate, Treponema pallidum can actually cause congenital syphilis as the microbe is motile and is able to cross the placental blood barrier. So there's mother fetus transmission, and it can cause syphilis in the neonate. So the treatment, the drug of choice for treponema is penicillin. So it looks like this in under the microscope. So you can see the corkscrew appearance and the flagella or the periplasmic flagellum. So here we have a highlighter. Okay, so here, so you can see the flagellum, a periplasmic flagellum, which is slightly contrasting. Okay. More about its morphology. Let's go back to the other slide here. Okay, so there's an outer membrane covering the cell. There's a periplasmic space in between the cell membrane of the cell and the outer membrane. Okay, the protoplasmic cell cylinder. So essentially, if you cut across if you do a cross section of a spirochete cell, so you can see that structure. And you can see the appearance as a gram-negative microbe. Okay. Now let's take a look at the clinical importance of Treponema pallidum infection. So this species can cause syphilis, and syphilis have different stages. Okay, so first is primary syphilis, which is characterized with the development of a painless canker at the site of infection, the site of inoculation, usually 3 to 6 weeks after initial contact. So the canker is a firm, ulcerated lesion that is not perceived as painful. It has a punched out base, so it's depressed, and there are edges that are rolled surrounding it. The canker is a highly infectious manifestation of primary syphilis as it is filled or is with concentrated cells of the spirochete. So canker looks like this here. So you can see the depressed appearance of the canker, and the edges are rolled out. And if you zoom in, if you try to get an oculum from it, you'll see that it has spirochete cells. Then there is secondary syphilis, which is the progression of the primary state if the patient did not undergo treatment. So characteristic finding associated with secondary syphilis is development of a condylatum, which is also painless, but the lesion now is wart-like in appearance, usually inhabiting warm spaces or moist spaces, moist sites with positive ulceration. And similar with the canker, it is packed with spirochete cells. So here is a condyloma lata. So it's wart-like in appearance, and it's filled with the spirochete cells. So associated with syphilis is rash in relation to the immune response of the host to the invading pathogen. Then there is latent syphilis when secondary syphilis is already resolved. In such cases, most patients are asymptomatic, and about 1/3 of untreated patients actually carry out or progress to tertiary type of syphilis. Tertiary syphilis is now the end game of syphilis infection. So it's the bacterium has gained distribution to other parts of the human host, characterized by very slow inflammatory damage to organs, to multiple organs and tissues, to blood vessels, and particularly targeting the central nervous system, nerve cells, the brain, and the spinal cord. And characteristic feature of tertiary syphilis are gumas, which are localized granulomatous, non-infectious, unlike canker or condylomas. Uh, the lesions are developed or are able to develop on skin or on the bones. And they're usually necrotic, causing the death of the tissue, and fibrotic in nature. So here is neurosyphilis, manifestation of the spirochete gaining the ability to invade nervous tissues, um, affecting the features that are coordinated by the nervous tissues. So Bell's palsy, neurosyphilis, characterized with imbalance or uneven appearance of the face. There's drooping of the mouth and the inability to close one of the eyelids. Okay, so gumas look like this. They're fibrotic in appearance and necrotic at the same time. So they can cause the death of the associated infected tissue. Then there is Borrelia burgdorferi, which is the agent of Lyme disease. It's a tick-borne infection. The animal reservoirs include vertebrates such as mouse, rodents, and deer. The vector of the spirochete is an exodus tick. And during early localization, so it appears as an erythema chronicum migrans. The treatment for this infection is doxycycline and penicillin. So here's the appearance of Borrelia burgdorferi. So um, you can see the um, cells, which are corkscrew-like in appearance. And here you can see the manifestation of Borrelia burgdorferi infection. Okay. So, what are the symptoms? They may have, Borrelia burgdorferi infection can is characteristically associated with what we call a bullseye patch rash. So it's a bullseye target-like appearance of rash. So here you can see that in this picture. The erythema migrans, I'm not sure if I have a picture. Yeah, here. So erythema migrans are migrating redness. Okay, so the rashes from um, the rashes actually erythema migrans. Okay, so it can appear as localized inflammation or localized redness in different parts of the human host. No? What else? In most cases, okay, 60 to 80% of patients with Lyme disease manifest erythema migrans. In terms of diameter, it's big. The the appearance of the redness about 5 centimeters in diameter. Then there is Borrelia recurrentis, which is the agent uh, or the causative agent for relapsing fever. It's transmitted by another invertebrate, by the Pediculus humanus, the body louse. Um, then we have Leptospira, which is another important type species for the spirochetes, the causative agent of leptospirosis. In terms of appearance, Leptospira interrogans is long, thin, and tightly coiled, usually appearing with an eye-stong feature or eye-stong appearance. There is hook on one or both ends of the cell. It's found in the urine of infected dogs or the infected carriers, rats, livestock, and wild animals. Let's take a look. Okay, so here you can see Leptospira interrogans, tightly coiled siya, compared to the other spirochete type species. So how does Leptospira interrogans gain access to the human host? So it penetrates through abraded skin or mucous membranes upon contact with contaminated urine, either by directly wading or swimming in flood water. That's why during the rainy season, it's part of the health promotion or health advice from central agencies, from health agencies, that people should avoid wading or swimming in flood waters, as the non-vertebrate human carriers may carry the pathogen in their body and secrete the pathogen through their urine. Okay? Now, clinically, let's take a look at the phases of leptospirosis. So we have the leptospiremic phase, which is when the pathogen invades the blood and cerebrospinal fluid, characterized with high spiking temperature, malaise and myalgia, muscle aches, meningisms, red conjunctiva, and the sensitivity of the host, the patient, to light, or photophobia. Then there is the immune phase, where antibodies against the pathogen are already appearing. So IgM antibodies are appearing. There's possible development of meningitis and associated liver damage, as well as renal or kidney failure. So the leptospirosis rash looks like this. It's macular in nature. And we in the succeeding stages, leptospirosis can become more aggravated where the disease is now characterized or is called given a specific name, so called Wales disease, infectious jaundice, primarily affecting the kidney and the liver. So can cause renal failure, hepatitis, inflammation of the liver tissue with jaundice. Can also cause hemorrhage in multiple organs, as it will now be more distributed. It will gain it will now be more distributed or has the capacity to invade other organ systems. So how do we treat leptospira infection? So we can have penicillin and doxycycline as the first line of antibiotics. So this is Wales disease. You can see the appearance of the patient with red conjunctiva and jaundice. Okay. Then we have the Rickettsia and Chlamydia, which are actually characteristically similar with viruses. They're very minute. They're obligate intracellular parasites, meaning they have to take over the host mechanisms or the host cell in order for them to survive. Okay. And they cannot be. It's hard to actually culture them in artificial media. Okay. So like bacteria, they have both RNA and DNA. They can synthesize their own proteins, no? And they can be cured with antibiotics. So for Rickettsia, they're rod-shaped in appearance or cocobacilli in appearance. They're motile and they can be transmitted via invertebrates such as insects and ticks. Their tissue of choice are the endothelial cells that line blood vessels. And some of the sample species and the disease that they can cause with associated rash are the following. So we have R. typhi, R. prowazekii, and R. rickettsii. So typhi causes murine or endemic typhus. Prowazekii causes epidemic. It's more adapted to population level distribution. And rickettsii causes Rocky Mountain spotted fever disease. Okay, so this is the vector of Rickettsia, and you can see Rickettsia cells stain red here inside associated host cell. Okay, and we have the Rocky Mountain spotted fever, which is spotted rash. The rash is usually absent on the onset and may not really appear right away. Okay, usual antibiotics that kill most bugs do not really affect this kind of infection. No, the rash moves centripetally and becomes hemorrhagic as the infection gains momentum, or as the cells, the bacterial pathogen, is able to thrive in the body of the host. Now we have the chlamydia, which are non-motile, but similar with Rickettsia, are coccoid in appearance. They do not have a peptidoglycan layer. Instead, what they have is cysteine-rich proteins on their membrane. They can be transmitted via interpersonal contact or via airborne respiratory route. Their tissue of choice are columnar epithelial cells lining mucous membranes. So chlamydia growing inside human endocervical cells. You can see the green colored cells, so they are inside the host cell. So it can exist in two forms during its developmental cycle: an elementary body, which is metabolically inactive but is infectious, and a reticulate or an initial body, which is metabolically active but non-infectious. So take note of the life cycle stages of chlamydia. Sample species include the following. So we have Trachomatis, Chlamydia trachomatis, causing trachoma, pelvic inflammatory disease, and urethritis. Trachomatis, you can see immature inclusion bodies, the elementary bodies and reticular bodies that keep on dividing, so they can produce more reticulate bodies. So trachoma, it's a type of chronic conjunctivitis which can lead to preventable blindness. Okay? Then we have Haemophilus influenzae, Bordetella, and Legionella as additional examples for the type for key species within the chlamydia group. So Haemophilus is blood-loving, hemo, then philus. Influenzae can cause flu, no? Pertussis, for the case of um, Bordetella, the woofing cough, violent cough. Then Pneumophila, which suggests that it is actually lung loving for the case of Legionella pneumophila. Okay, they are primarily acquired via airborne respiratory tract route. Haemophilus influenzae can cause respiratory disease, infection of the infection of the throat structures, like the epiglottis, and may also cause meningitis. Woofing cough is caused by Bordetella pertussis and Legionnaire's disease, which is actually a very recent what do you call it? It's a very recent disease identified, I think, in the 80s or 90s. No, Legionnaire's disease. Okay. I think that's it. We end our discussion on the medically significant gram-negative bacteria. There are two more recordings I will complete on endospore-forming bacilli and the non-sporogenous bacilli.